Hola, bona tarda. Good afternoon. Welcome to the session Biotech, Democratizing the Practice of Biotechnology. This is a session. For this session, we come with four fantastic people. We have Mark Way, who is a group leader of the Translational System Biology group at the Department of Experimental Health Sciences of the University Pompeu Fabra in Barcelona. Then we will have Tom Peters, a senior scientist at Erasmus Brussels University of Applied Sciences and Arts, and he was as well a founder of the Open Bio Labs Brussels. Then we will have Núria Montserrat, an ICREA researcher and group leader of the Pluripotency for Organ Regeneration group at the Institute of Bioengineering of Catalonia. And finally, we will have Núria Conde, who will join us in a little while. And she works at the Complex Systems Lab at the University of Pompeu Fabra. And she's one of the founders of the Biohackers movement in Barcelona. And I will be chairing this session. I'm Cristina Pujadas. I'm full professor at the Department of Experimental and Health Sciences, as well at the University Pompeu Fabra. And just for you to know how the session will run, we will have the 4 talks of the speakers of 12 minutes. Afterwards, we will open for a dialogue, a conversation among the speakers that will take more or less 15 minutes. And then we will open the floor for you. And you can send questions, and we will bring them to the speakers, and they will respond or they will dialogue or conversate around these things. Therefore, without further delay, I'm going to pass the floor to Mark Wayne. And Mark, whenever you like, please.
Okay, excellent. Thanks, Christina. So let's see. I think, okay, I think you should be seeing my screen, right?
Yes.
Okay, excellent. So yeah, so first, yeah, thanks a lot for the invitation. Very happy to be here with so many friends and colleagues. So yeah, so today I will discuss mostly about how to basically what we do in our lab in terms of applied synthetic biology. So I will talk mostly about mammalian genome engineering and a little bit about skin microbiome engineering. So first, I would like to make a small advertisement. So a big shout for the IGEM team of the Barcelona IGEM team. They have really great results in the IGEM competition. For those that you don't know, it's one of the big world in synthetic biology. So we have a good future generation of makers, at least in the symbio field, for the next years. So a big shout for them. Okay, so today, yeah, as I said, I will discuss a bit some tools that we have been developing to do precise gene delivery in human genomes. And then at the end, if I have time, I will discuss a little bit briefly some tools that we are developing to engineer the skin microbiome, so these bacteria that we have in the skin. So I'll start. So I think it's not a secret that we had this huge revolution in biology. So first at the RIP level, so we can sequence very fast genomes, but also at the bright level. So we have been synthesizing full genomes from scratch, bacterial genomes, synthetic, even eukaryotic genomes like full yeast chromosomes. And yeah, I'm sure you have heard in the news this big revolution on the editing field, especially thanks to the CRISPR machinery that allows us to do these fascinating edits on mammalian genomes and prokaryotic genomes as well, actually. So this has been very impactful, of course, in the basic science field. So it has been used in, I think, all the model organisms, and also it's translating very fast to the therapeutic part. This enhanced capacity of engineered genomes, well, it's already in the clinic. Here, I'm just showing a list of numbers of clinical trials, which it's in the order of thousands. So really, genome engineering has accelerated the discovery, but also the translation in novel therapies. So today I will speak of some tool that we have been developing in the lab, which is basically a tool that tries to combine this precision of CRISPR, but also with some more classical capacity with this, which is gene transfer, some tool to do precise gene transfer in mammalian genomes. So normally when we talk about genome engineering in mammalian systems, we usually use CRISPR or another tool to do a double-stranded break, and then we induce this system to repair either by novel joining and get some indels, and in this case, inactivate genes. We'll see that this is very useful, actually, as we heard this week in the news, that's very helpful for patients already. Or we can also induce repair using homologous recombination and generate new alleles. So CRISPR has been you can also use CRISPR on other different ways, which is not only double-stranded breaks, which is, for example, the case of base editors, where the CRISPR, the Cas9 protein, the main CRISPR component, can be attached to, for example, a deaminase, and basically generate a tool that can change Cs to Ts, or sorry, this is a mistake, actually, or As to Gs. Also, even more recently, people have combined CRISPR with a different system, which is a reverse transcriptase, where you can have not only base change, but also basically base replacement. So directly here, you put a reverse transcriptase where Cas9 brings in the genome, it can synthesize another piece of DNA which gets incorporated. So then not only you can change bases, but you can replace bases, you can basically make insertions, deletions. In this case, always small fragments. Some of these tools, especially the double-stranded breaks that I showed before, they have already entered into the clinic. This was like some data announced last year on engineering T cells. But also very recently, we saw that actually also in the field of hemoglobinopathies, so basically in sickle cell anemia and beta thalassemia, there were also very good results. This is actually some early results that we saw for 2 patients. You see the stock of the companies sponsoring the trials, how they jumped high. But this two patients' data got confirmed this week. It was presented in the ASH conference with, I think, it was 9 or 10 patients. So using CRISPR in this case to wake up the fetal hemoglobin by breaking the regulator that keeps it dormant. And yeah, this, it seems that it has been one of the first successes of CRISPR therapy. Not only showed safety, but also it seems very, very good efficacy. However, so it seems that we have good systems to do basically a small allele edits, either breaking alleles or to insert new alleles by the prime editing or base editing. But actually, and this, so gene disruption, we saw it's very efficient. Even we can generate efficacy data. Base editing and prime editing seem promising for correcting alleles as well. But what happens when we want to do gene delivery? What happens if we want to insert a full gene? So in this case, traditionally this has been done with retroviruses. So we know from the CAR-T field that basically people have used lentiviruses or gamma retroviruses. And also we see that CRISPR, it's true that it has very good properties, but also it could generate some this we can discuss later, or even from the point of view of generating, for example, bronchogenic behaviors. So what we have done in the lab is we have tried to combine this precision part of CRISPR, which is basically finding these specific places on the genome, with some kind of enzyme that can do an insertion, which is basically a professional gene transfer model, what we call this cut and paste model. So, and with this system, we can get an efficient delivery of large payloads. Of course, we can be precise because of the Cas9. And then also by having basically this by not having this dependence with the DNA repair machinery, so we can be a bit more universal and target cells that divide and cells that do not divide. So this is the system, how it works. So it scans, it finds a place, and then it performs the insertion. We could show that this system works quite efficient. So it can be almost as efficient as a non-programmable system, a classical transposase. Another good property is that the scales very well with size. So we can, since the transposase basically helps to insert the two ends of the DNA, so it can be very efficient even for large sizes compared to the, let's say, classical CRISPR gene insertion. And then also we can be quite precise. So this is the system before engineering. You see all the red dots. Here we are targeting the TCR receptor where the green dot is. So you see before engineering how many red dots we have. However, after the engineering, we get this very precise system that all inserts in the TCR receptor in this case. This system works well in mice, so we can measure good copy number after some time. Here I'm showing also some luciferase. We have tried to use this system for genetic disease. In this case, the first indication that we tried was a congenital muscular dystrophy that is caused by mutations in the laminin gene, which is a very large gene. That's why we choose this disease, because actually it's very difficult to tackle with more traditional technology. And then in this case, we can perform the gene targeting and really bring the lifespan of mice to the almost normal lifespan. And also we can make them increase strength. So yeah, this is two more cohorts of mice where we can increase this lifespan by the gene transfer, this precise gene transfer. So as a summary of this part, so we could obtain this programmable transposase, which works quite well in terms of efficiency. We believe that this could be a quite safe system because we have a very controlled way to perform gene editing. And then, yeah, we could show these early phenotypes on this muscular dystrophy caused by mutations in the laminin-1 gene. Laminin 2. So in the last 2 minutes, I would also like to discuss a bit some effort that we are doing, not engineering directly mammalian cells, but engineering the prokaryotic cells that live with us, the prokaryotic cells that live in our skin. So you know how important is the microbiome, right? How important it is for our physiology, how important it is, and also how fascinating molecular machines bacteria are, right? They are really fascinating molecular machines that we can engineer them to do very interesting things. So we can engineer the microbiome from the point of view of changing the composition, right? This we have tried since quite a while, and this is a bit inspired on past, for example, efforts in doing fecal transplant, in changing the microbiome of the gut, for example, to address, for example, Clostridium difficile infections. This has been very efficient. So we have tried since several years to do this in the skin to modulate, for example, the composition, to turn it into a more basically to break some dysbiosis, so basically to turn into a more healthy state. In this case, we have been doing it in the context of acne since quite a while. But the first question is, can we change this microbiome at all, right? Because and the answer is yes. We can produce basically probiotic solutions that they really stay in the skin. In this case, with very simple testing. So on healthy volunteers, basically, we prepared several probiotic solutions, apply them into patients, and then actually we see that they last quite long. So especially certain compositions, so we can make them last for actually several weeks. But is this efficacious? So this, okay, we can change the microbiome, but this is useful. So we did some early and very small clinical trial a while ago. And in this case, we could demonstrate that we see, for example, a decrease in the inflammation in the skin in the context of acne. Of course, here you see the numbers. This, because of lack of funding, these numbers are very small number of patients. Now this is being repeated in collaboration with Bayersdorf, a big pharma company in the domain. Big trial. Hopefully, we will see how the data looks. So from what is a summary of this part? So these probiotic solutions are very safe. Actually, we can really change long-term the skin microbiome. And then finally, we can observe some clear, well, at least some preliminary positive trends in terms of phenotype. But actually, we can go beyond. Actually, we can change this microbiome. This is a big opportunity. It's a big platform to add functionality into the skin. So from a synthetic biology point of view, so adding new genes that do not exist. And in this case, it has already been tested in some contexts using genetically modified microbiomes, even in the skin. This is a pioneering company in Connecticut working in Netherton syndrome, and also in Europe. We have had efforts, very interesting efforts, for example, in producing very short-lived cytokines on the skin for, in this case, bone healing. So in our case, what we are trying to do, so we know we have this very strong platform of modifying the components of our skin microbiome. So during these last years, we have developed reliable methods to genetically engineer these microbes of the skin, which actually they are very hard. So this was a quite difficult project, but now we have these good methods. And then we are teaching these bacteria to listen. So basically, we are creating biosensors so that we can listen our body, our sebum secretion, cytokines production, and then teaching them to actuate. So basically, create genetic circuits that, based on this state of the skin or our environmental exposure, to do different things. And yeah, and actually with that, I would like just to thank my lab, and yeah, that's everything I had today. Thanks a lot.
Thank you very much for this fascinating talk. Now we will pass the floor to the other speaker, no? Tom, whenever you like, and we go all the four in a row, and then we will open the discussion, okay? Thank you, Tom, whenever you like.
Hello, can you see my screen that I shared?
Yes, we can.
Okay, so thanks to the organization for inviting me. So I'll give a short presentation about the Open Biolab in Brussels and about some of the projects that we're doing there. So, but let me start with explaining briefly what DIY biology or do-it-yourself biology is. So do-it-yourself biology, it's a global movement. So that's important to know. It's a global movement. It's a diverse group, but they all have a shared goal, and that is to make biology and specifically biotechnology accessible for everybody. So once a year since 2017, DIY biologists group together in the MIT Media Lab to share results and discuss this movement. And they also set up a shared purpose because, of course, one of the things that they're focused on is biosafety. So it's a diverse group, and some people are doing weird stuff, I must admit that, but most of the people are really know what they're doing and want to make biotechnology accessible and in a safe manner. So when I got introduced to this movement, I thought, okay, this is something very cool. And since I teach to lab technicians, I thought, okay, we have to start doing this in our school as well. And we started a lab that we open to everybody. So also people that are not students are welcome in the lab to start experimenting. So we have a lab, of course, but if you don't have a lab, then there's one thing you can do. That is to build a lab yourself. And then, of course, it costs a lot of money. So what people started to do quite fast is to make their own lab devices. And this you can see here. This is something that's developed by Waag in Amsterdam. So they made a lot of equipment and share everything on the GitHub. And of course, they were not the only one, but other players started as well. And for instance, this is some interesting device that we also bought. This is a machine to copy DNA, a PCR machine, and you can buy it for $500 from OpenPCR. So it arrives like an IKEA building package, and then 3 hours messing around with a screwdriver and then ending up with 5 parts, of which you don't know where they belong, but then you have your PCR machine. And you can even go next level. As you can see in this article, what researcher did with some microbits and then some soup cans and some glue and metal wires, they made their own PCR machine. So this is a small robotic arm that puts the tubes, as you can see, small tubes from one can to the other. And that's how you can also start PCRing. They even published this, so this works, which is extremely nice to see. So when I saw this, I thought, okay, this is something that we should do as well in the lab. And I got some small funding from the city of Brussels, and we also made a PCR machine ourselves. So this PCR machine works, but normally PCR, it takes 2 till 3 hours, and this took 8 hours. So it works, but it works slow. And then we also made a gel electrophoresis device that you use to separate DNA. So when we made this stuff, I was very proud and very happy. But then I met Cristina Cipriano from Italy. And what she did, she made a gel electrophoresis device from a Tic Tac box. So this is really next level. You cannot beat someone that makes gel electrophoresis devices from a Tic Tac box. So we also made a centrifuge, and this we made pretty because, as you know, in most labs, lab devices are always like this grayish white color, and they don't look nice. So we made a funky red centrifuge to spice up the lab, and we also made some renders of other devices, which we didn't build in the end, and which I hope we will build. But we made a Metallica themed DIY centrifuge, or a very posh one with the Louis Vuitton team. So you can make lab devices also interesting to look at. And one other device that we developed was a DIY incubator. So this is a device that you need to grow cells, and in this one you can grow microbes. And for this, we organized also a 5-day STEAM workshop for youngsters in Brussels. We had 22 participants, and we were very pleased that 17 participants, were girls from the city of Brussels that partake in building an incubator in 5 days. Then together with the graffiti artist, they sprayed it according to their taste. And then in the end, we also displayed those in the big library in Brussels, so everybody could see them and see how they worked, and got some information about these devices. We really hope to organize a second edition of this, and I'm currently working on finding the finances for that. Because with this device, you can start growing your own bacteria, and for that, of course, you need a bacterial growth plates. And for that, we developed a STEAM kit, which we called Let's Swap. And with this kit, you learn how to make your own growth plates with kitchen devices. And one of the things you have to do is to sterilize everything. And for that, we used an IKEA cooking pan for 60 euros, and it works fine. And here you can see a plate of a medium that we made with things that you just buy in the grocery store. And we grew several bacteria on that plate, and you see it works well. So very nice kit, especially for Brussels schools. So we used it for Brussels schools so they can, with low cost devices, start making their own bacterial growth plates. And then, of course, to make it more interesting, or to make it more interesting for youngsters, we also competed in the Agger Art Competition. And this is a competition where you can make drawings with pigmented bacteria on these growth plates. And here you can see some examples of things that we've done. So very nice mandala made by a student of ours and a minion, and then I made this clown for my daughter. And underneath you can see this pic that our Red Devils, our soccer team, we made that for them, for our national soccer team, to encourage them to win the World Series, but in the end they didn't. So, but very nice. This is something that's how people get acquainted with biotech, with microbiology in this case, and on an interesting and funny way. Also, so we have a lab, and this lab is open. Well, now due to COVID, it's closed, of course, but it's open every Wednesday afternoon. And then we organize several workshops. So here we're training some teachers on how to use our Let's Steam kit, Let's Swap Steam kit, so they can grow their own growth plates. But we also go out to science fairs where we show what we're doing and organize small pop-up workshops for youngsters, so they get acquainted with biotechnology, or at least have a pipette once in their hands before they start studying biology, for instance, when they're 18 and they never ever hold a micropipette, which I believe is extremely silly. But it's not only education, or not only fun stuff. I think that's very important to make people interested in biotechnology. But just recently, we also started with a developmental project in Peru. And here you can see the Nanay River in Peru. So it provides water to over 500,000 people. And on that river, there's a lot of illegal mining. So there's gold in that river, and people in those boats, as you can see in the picture, on those boats, they suck up all the water to clear it, to look for gold particles. And to catch the gold particles, they have to use mercury. And of course, they don't know how to handle that properly. It's also very illegal. And the mercury gets back into the water. It's get taken up by the fish. The people eat the fish and people get sick. So to make people aware of this problem, we started to build biosensors. And this is something that's funded by the Flemish government, by FAIRE US. And it's a collaboration with Universidad Peruana Cayetano Heredia in Lima, and we made whole cell biosensors. And here you can see a graphic of what a biosensor is. So that's a genetically altered cell, and when it senses something, when there's an input, this input through a genetic circuit will get into a signal. And what we built are E. coli cells that, when they sense mercury, when they get in contact with mercury, they develop this very nice blue color. So this is a device that people could use. So we're now setting up a whole project in Peru where we will train teachers on how to use this device. And so people can check water themselves and see if water is contaminated with mercury. So that's my short presentation. So you can follow us on social media, and I encourage you to specifically check our Instagram, because then you can see with what we're doing.
Okay, thank you very much for this nice presentation, Tom. We will come back to the very same movement in the last talk with Núria Conde, but now we are going to pass to another way of dreaming. It's as you dreamed with Mark, all the things you can do with CRISPR and microbiota, etc. Now Núria Montserrat will tell us all the things you can dream to do in printing. Thank you very much, Núria, whenever you like.
Thank you very much for the invitation to this wonderful fest. and I hope to be clear with the presentation. I try to bring the slides from the lab, the work we do in the lab. We work with pluripotent stem cells. And let me go first. I don't know, now it's not working. Yes, so let me go first with this slide that goes very back on the history. So this first slide represents how the Egyptians were already talking about a fire bird that was called Binu, this blue bird, that was having the capacity to birth all the time, so every season. And for the Egyptians, this bird was representing good things like having more food to eat and also to have a period of fertility. This myth was then adopted by the Greeks, known as the phoenix, and nowadays we are very well adapted into this, but I think that this myth recapitulates very well the major goal of regenerative medicine, which is to restore, regenerate, or replace damaged tissues. And this is how I will try to bring you into a journey in where you will see that most of the things that we are now listening, for example, stem cells, and many other works that we are listening very lately on related to biotechnology are coming from very old studies in biology. So let me also remind you that in nature, there are animals that can regenerate. So they are suffering an insult, for example, and these animals can generate tissue de novo. This slide in the center, you can see the structure of a nephron, which is the functional unit of a kidney, and small fishes and also amphibians can generate nephrons de novo whenever there is a harm. Also, in the laboratory, we work with animals that they do have this spontaneous regeneration or this endogenous reparation, as the zebrafish, in where you can manipulate this fish and amputate, for example, the 20% of the mass of the ventricle of this fish. And this fish, in a period of 20 days to 30 days, will regenerate the damaged tissue. And also in the lab, we are working with embryonic tissues. And why is so is because tissues have the capacity to self-organize and to reassemble in the Petri dish without providing much more effort, but just providing few instructions. We can, for example, culture in vitro kidneys from mice during development, and these kidneys will be aggregating again and forming de novo after dissociation, and we can monitor in the Petri dish how the cells are going to be assembling this kidney. All these analyses and all these studies are not new. I'm always wanting to talk with young people to tell them that these studies were already ongoing at the very beginning of last century. Already by the beginning of 900, Loeb was taking advantage of rabbit embryonic tissues to study this kind of phenomena. And by the 50s, Moscona and Moscona, they were also disassembling or reassembling chick or mice embryonic tissues and studying all these processes that now in the lab we are taking advantage of. But it's true that we are taking also tools that are more evolved, like using transgenic animals, but still the questions are there. So how tissues assemble, how tissues are formed, and from where distractions are coming. So regenerative medicine is a multidisciplinary field in where we are taking advantage of these animals that have the spontaneous capacity to regenerate, but also from stem cells and therapy, as Mark Well was telling in his first talk. And also in regenerative medicine, other disciplines that are taking place are related with the use of human tissue engineering. And in my lab, in IBEC, we are taking advantage of stem cells, but not, I will also like to tell you a few words about the stem cells. So stem cells are found already in our bodies when we are adult. We refer to these stem cells as multipotent stem cells, as the ones we find in the blood, for example, hematopoietic stem cells, or unipotent stem cells that we can find in sperm. But during embryogenesis and during human development, from day 5 to day 6, during human gestation, we can derive and we can find human embryonic stem cells that we can isolate in the laboratory, and we can culture, and these cells are known as human pluripotent stem cells. And these cells are the ones that have the capacity to give rise to all the tissues in our body. And I will just go very quick into this slide, but I think it's very nice to see, especially for the young audience, that all these questions are here for a very long time. So already before the last century, there were already attempts to fertilize oocytes in rabbits to understand how this process works. And of course, maybe some people in the audience that is older as me, we all remember the first successful FIP in humans by 78, the year I was born, by the way. So already by the 90s, human mouse embryonic stem cells could be derived in the laboratories, and a seminal study by 98 described that it was possible to isolate embryonic stem cells from human embryos, specifically by dissecting this small clump of cells that is called inner cell mass, in where we find transiently human pluripotent stem cells in human embryos. And for more than 20 years, my laboratory, but many labs in the world, have been addressing how to culture these cells in the Petri dish without losing the properties of being pluripotent. So meaning that these cells, eventually, when we want, we can instruct them to become whatever cell we aim to derive in the Petri dish. Another seminal study and another big step in the field of stem cell biology was done by Professor Shinya Yamanaka, who was awarded in 2012 with the Nobel Prize for Medicine and Physiology, as he discovered for the first time that the overexpression of 4 factors that nowadays are known as Yamanaka factors could generate in the Petri dish cells that were very similar in characteristics to embryonic stem cells in the embryo. And these cells were called induced pluripotent stem cells because the pluripotency state was induced artificially in the laboratory. So nowadays in the lab, we do have access to human embryonic stem cells from embryos, and also we can derive human iPSCs, human-induced pluripotent stem cells, taking advantage of Yamanaka factors, but also by other factors that we have been taking advantage in the laboratory. And something that I will be very briefly commenting in the last 7 minutes is that in the lab, we are very focused in understanding how to control the differentiation from undifferentiated pluripotent stem cells into tissue-like structures, and that we are taking advantage of the self-organization capacity that the cells possess to generate mini organ-like tissue structures in the Petri dish. And why we are doing this big effort is because we want to understand how tissues develop, to understand eventually how tissues get disease. But because we are facing here a big challenge and we need to derivate tissue surrogates, we are taking advantage of bioengineering strategies in order to further maturate or differentiate these tissues. So in the lab, we were successfully generating mini structures that were resembling the kidney of the second trimester gestation in human, and these micro kidney organoids were teaching us important steps during the kidney differentiation in human. And then taking advantage or using hydrogels that are mimicking the stiffness of the tissue, we were showing that we could better generate these microorganoids, these microkidneys, and that these microorganoids generated in a soft microenvironment, resembling the microenvironment and the physical cues that the cells encounter in the tissue, were better differentiated. But bioengineering can also afford and make us become or make more things. For example, in the field of tissue engineering, already by the 70s, it was shown that we can take advantage of a whole tissue of organs, and we can remove the cells of the organ and work with the scaffold of the organ, which is composed mainly about extracellular matrix proteins. And these proteins are known to become or to behave as instructors of differentiation of the cells. So in the lab, we have access to discarded human kidneys and also porcine kidneys. And we are delivering or generating our own scaffolds for performing our experiments. And also we can take advantage of these scaffolds to generate hydrogels for applications, as Christina was mentioning, in 3D bioprinting, as these scaffolds are going to be allogenic and universally used, as they don't possess any more DNA that will give rise to rejection in a context of transplantation. So we are working in the derivation of these biomimetic hydrogels for the generation of these organoids. We are taking advantage of these substrates for generating even bigger and more mature structures, profiting from these instructive signals that the ECM or the extracellular matrix already has in the cells. And we are scaling up our procedures of differentiation for generating organoids, taking advantage of 3D bioprinting. Something that I think it's also relevant to talk about, although we are all very tired about COVID-19 disease, is that organoids can be very amenable to understand, for example, disease gestation progression. And something that came into us very quickly already by February is that we were aware that SARS coronavirus 2 was using as a putative receptor ACE2. The angiotensin converting enzyme ACE2 is not only expressed in the epithelial mucosa in the nose or in the lungs, but also in the kidney. So the first thing that we did by February was to profile using transcriptomics our kidney organoids to understand if these microtissues could be amenable to understand the first steps of infection of this SARS coronavirus 2 infection. And as you can see here in red, a small proportion of cells within the kidney organoids were expressing ACE2 in a similar fashion as we observe in a native kidney. So what we did in collaboration with Josef Penninger in Austria and Ali Mirazimi at the Karolinska Institute was to try to understand if we could use these microorgans to understand these steps of infection and also to profile compounds to see if we can block SARS coronavirus 2 infection. So this is something that I think it's important to recall. We have been doing this in a context of lockdown, but thanks to work in collaboration, we could ship our kidney organoids to Sweden every week. And the group of Ali Mirazimi, they were infecting our kidney organoids, and they were observing how the virus was infecting these kidney organoids, and we were processing back the samples here in Barcelona. And the first thing that we observed collectively was that a compound that was described to be safe for safe patients or normal population, and also used for treating pulmonary disease, was blocking or inhibiting the infection of SARS coronavirus 2 from 1,000 to 5,000 times. And now this compound is already being tested in COVID-19 patients in 6 countries all over the world. And I think I just have, Christina, how many time left?
No, you are doing pretty well. You still have 3 minutes.
Very few, 2 more slides. Something that I think that Mark Way has been explaining very well is on the application of CRISPR-Cas9 technology in somatic cells, but human pluripotent stem cells are very amenable for genome editing applications, as we can culture them in single cell suspension, for example, and they can be transfected very easily, and we can select these cells for further applications in genome editing. Now is nowadays in my laboratory. When he was working in Sloan Kettering, he already targeted a safe carbos locus in these cells for the insertion using TALEN for the insertion of a module for the expression of Cas9 under the TETON system. So this design was occupying the 2 allele of this ABBS1 safe carbos locus. In human cells, we do have other locus, but we were using this because it was shown before that targeting this locus was not harming or affecting the differentiation capacity of human pluripotent stem cells. And now, in these last 5 years, we have been working a lot in repurposing this scheme for targeting human pluripotent stem cells. And now we have heterozygous or homozygous pluripotent stem cells, including human embryonic stem cells or iPSCs, in where we can induce the expression of Cas9 for applications, for example, in generating knockouts or knock-ins, and also using that Cas9 form, we can also upregulate or downregulate the expression of specific target sequences that we want to understand how they are impacting on development, but also in disease. And this is one example, as I think we are very familiar with this, but it's important to recall that by doing these modifications, we can now screen for a lot of different guide RNAs and test, for example, on the efficacy of targeting for a specific loci that are important for development, for kidney development, for kidney disease, and for processes that are important for cell biology, as for example, dicer. And we are taking advantage of this platform to understand, for example, how specific mutations or deletions in genes that are important for development are compromising kidney differentiation, and even using these organoids for screening for compounds. So this slide is more for a generic conversation, maybe after our 4 talks, for stressing that we can go from very basic questions in biology to translation at the very end of the road. And this is taking a lot of time and a lot of effort, but we need to always take into consideration that multidisciplinary approaches have been very useful in biotechnology. So the question here for us in the laboratory is, how can we advance this progress? How can we make this a reality? And also how or which ethical and social questions is our research implicating into the society, because we are working with pluripotent stem cells. We are taking advantage of cadaveric tissues for deriving our hydrogels and for building these structures de novo. So I would like to devote 30 seconds. I think this is the most important slide of my presentation today. I would like to thank the people who is doing all the work in the lab, the collaborators for this SARS coronavirus 2 project, and also for all the funding agencies for allowing me being here today. And thank you very much for your time. Thank you very much.
Thank you very much, Nuria, for driving us to this new way of prediction medicine that you just opened here. And we will discuss a bit about the ethical issues in the further questions. But now, after thanking you, let me pass to the last speaker, Nuria Conde. Nuria, welcome here. And
welcome.
And now it's your time. You have 12 minutes to tell us or to share with us what you wanted to tell. Thank you.
Can you see my presentation and listen to me?
Yes, very well, thanks.
So first of all, thank you the organization for inviting me, especially for the honor of being with the rest of the scientists that are part of this panel. And I'm also a young scientist, but I'm here today to talk what I do after I left my professional laboratory. In 2014, I finished my PhD, and before jumping to the next step of the scientific career, that's doing a postdoc, I decided with a group of friends to make an open laboratory in a garage. We founded one of the biohacking labs that it was the first in Spain and the first in Catalonia, here in Barcelona. We come up with a super original name of DIY Bio Barcelona. And the aim of all of that, and the reason why I did that, is because I sometimes got these super amazing conversations like the ones that Mark and Tom and Nuria already shared with us, but I got also the feeling that the normal population that is not so near to science were really far away of understanding that the future of making a mini organ or changing a microbiome are already here. And as William Gibson said, the future was already here, is already here, and it's not evenly distributed. And the aim of doing an open laboratory for the citizens is just like open this black box in the similar way that already happened with the software when Linux appeared, or with the hardware when Arduino appeared. And the idea was to do the same with biology, and then distribute through the open source communities this knowledge that has been like locked on some of the laboratories that we work every day. That is our normal working day. So I started, as Tom explained it, to understand a little bit what is this movement that's called DIY bio or biohacking. And I just wanted to present to the audience that normally when you walk through the door to one of these biohacking spaces, you will find inside some people that are biologists, of course, but there is also engineers, normal citizens that just pass through the door like that, artists and makers. And when you go in, you will see people that's doing software, that's doing hardware, that's doing microbiology, biochemistry, biotechnology, that's actually also doing some kind of genetic engineering through one of the worldwide contests of doing genetic engineering bacteria, the Agger Competition. And mainly the activities that we do and the areas that awaken an interest to the people that has been working in our space is like teaching biology, that's what I do, but also a lot of people that's very concerned about the environment, their own health, about biotechnology applications, or even to do with the lab machines as Tom explained it. It's an open space that normally works through the internet, and it's everything that's done is open source. So we try to publish all the results and all the classes online for everybody to be able to access for free. That's the philosophy, and I think that slide kind of sum up what has been my hobby for the last 6 years. I also like to explain that even this sounds like a little bit dangerous, or at the beginning you think, am I crazy to become a biohacker? But then I studied a little bit of history, and I want to say that we are not the only biohackers. This is an idea that I like to say that's vintage. So before the science was inside of the big institutions in history, it was done mainly in the homes of the scientists that got someone, that the mecenas, or someone that pay a little bit for the research. And some of the pictures that you can see in the slide are, for example, the roof, in which you can see the electricity from the house connected to a frog. This is the original picture that the Galvani doctor or scientist published, explaining how the electricity can make a muscle to move. And the legend says that when he published that, a lot of people went to the market and the frogs were exhausted for some weeks because the people wanted to see by themselves. It would be awesome that that was true. Also, the picture that's in the middle is from Ramón y Cajal's bio. It's the first microscope with the camera that they will be using, and some of the neurons that he was discovering. And it was interesting when I was doing that research that I learned in one of the interviews, the last interviews that he did, that he was confessing that for a period of time he was afraid that his neighbors accused him of neuromancy because he spent a lot of time in the house, in the downstairs of the house, trying to come up with the mixture, the cheapest mixture possible to develop the films, because he was actually doing a lot of pictures of the neurons. And the last picture is a truck, is a Ford truck. The owner of this truck is Marie Curie. Is when she decided that the soldiers that were like dying and having injuries on the field needed the X-ray machine to be there. So she hacked a little bit what was being doing and reduced it and put it in a truck. And the last pictures that you see in the bottom, they are the machines that Rosalind Franklin used to make the first picture of the DNA. The very last one that you cannot distinguish of anything, it's some plaques that she was making of clips and gums and tape and everything that she can collect it around the lab to prevent the leakage of the helium that was needed to have a stream without turbulences, but some leakage were happening. So she was trying to patch up everything successfully because she come up with the picture. So with that, I want to make the message that even it sounds a little bit weird that you can do a lot of experiments in a laboratory or with machines that you do yourself. This is actually something that has been done in the past with really high success. It's to encourage the people to say that we don't do second line science. It could be first line science for if you really put the effort and the time. We are not alone. So the movement, as Tom explained it, with the picture of BioSummit is around all the globes. I got here, I only put the logos of the other open spaces that I have visited around the world, like Wax Society in Amsterdam. They help us a lot to start the one here in Barcelona. And again, I want to explain a little bit what we have been doing these 6 years here. So in one hand, to reduce the costs and to personalize the machines of the laboratory in order to have a laboratory to make experiments. So we need to make the machines, and we call some makers to do that. And then we did some experimentation, mainly in biotechnology and medical auto-exploration. As an example, these are not machines that we did, but these are the classic machines from the movement. Cathal Garvey, one of the biohackers that I met, it's the father of the small centrifuge that you can use a 3D printer, as you can see in the image, and then put this 3D printed plastic piece in one of the drills that you have at home, and then you can have a centrifuge for these tiny tubes of the laboratory, the Eppendorf, that it's not the same that the one that you have in the image, like the professional one, but for many experiments, it's more than enough. Also, you can see like a spectrophotometer. That's a machine that normally it's kind of big. It can weigh between 1 kilogram and 1.5 kilograms, and it costs around 1,000 or 2,000 euros if you want to buy one professional for the lab. Hacteria, that's one of the groups that I met around Europe that was doing DIY. They come up with one that can fit in your hand. The little coin that you can see in the image actually is a battery, a small flat battery. And it has been used by the farmers of Indonesia to check if the water that they are using for the fields are contaminated with phosphates and nitrates, also to assess in which concentration. Again, these ones, yes, this is something that as I said, I wanted to go in the direction of auto-exploratory medicine or to know a little bit better of yourself. And since my PhD is in DNA, the first thing I did is just look to buy a little mini Pcr machine to amplify DNA. I did it from one of the first startups, and now it's a well-known enterprise, Mini PCR, that started as a biohacking lab and then developed the machine, and now they are selling it. What we do is just like help the people to extract a little bit of their own DNA and then do an amplification of a funny gene, like the warrior gene or the gene that tells you if you got a taste for the things that are bitter. I didn't use a Tic Tac box for doing the electrophoresis machines. I normally use the boxes that for poker cards boxes, as you can see in the image upstairs. And then another enterprise that has come from a biohacking lab is the IO Rodeo. And we bought the transilluminator. That's the machine that allows you to see the DNA after the electrophoresis is done, and it also fits in your hand. So I can wear all my laboratory in my backpack. This was, of course, not something that I come up as myself. The first girl that come up with that idea of having a laboratory in their own cupboard was Kay Alul. I don't know if I can pronounce her name correctly, but she found out that his father got a genetic disease, and she wanted to know if she was a carrier. She was a researcher like us, and she decided to do all the laboratory at her bedroom and check if herself was a carrier or not. As you can see, this is the most typical image that you can find internet about that history. And with all this inspiration and with help of a group of artists that they are called Ginepunks, one of the first projects of the DIY Bio Barcelona was to make a kit for auto-exploratory gynecology. It's a kit that allows you to check some few things and see and take a look inside of your own uterus. And this is the picture of all the kit and all the classes that you take to be able to do this to yourself as a girl. We also do a lot of growing of bacteria, as also Tom explained it. As you can see, the first thing that we do of growing microorganisms is to make beer. We are a Mediterranean country, and that was mandatory. We also grow a lot of bacteria that's around. There is a very famous or very successful workshop that I do that's about hunting microorganisms around you. I normally don't use the normal Petri dishes, so I just ask the people to bring their own jars of small marmalade jars to put the medium inside and grow the bacteria inside, or even these plastic chupito glasses that you can buy in any disposable shop. I also have to say that hunting bioluminescent bacteria from the squids of the supermarket is also something that the people likes a lot, and then you can see how the bacteria glows in the dark. And another favorite, it's the Acetobacter chilinum. It's not a very well-known bacteria. For us, it's not commonly known as Scooby or kombucha. And as you can see, I made a little machine with Legos that can keep turning a piece. And then if you grow the bacteria on top, you can have that piece with a nice, not even, but a skin of bacterial cellulose. This is interesting for a lot of... Wax Society was the one that sent me the strain. As you can see, once you have this bacterial cellulose, you can use it as a material for making gloves or making clothes. But also with some students of Elisava and some architects and designers, we do a lot of, they like to come and do a lot of tests using this bacterial cellulose, also with another biomaterial that comes from crabs, that's chitosan, and also a lot of what they call oyster mushroom for the Catalan people, that's chircola. And this mushroom can make a lot of fibers, and they study the mixture of these materials to come up with new materials that have new properties. And as you can see, I teach them how to do the experiments and how to come up with new recipes that have different, as a result, different materials with different properties.
Nuria, sorry, you should be wrapping up.
This is the last one. The last thing that we did, or the most important or famous one that we did, is the one that we exposed at CCCB, and it was about bioprinting. It's just like printing this agar with the 3D printer, or with the, as you can see in the image, the cellulose of this bacteria that has been smashed, and then you can use the syringe for printing. And thank you for your attention, and then I give the word to Cristina.
Okay, thank you very much, Nuria. Thank you very much, Nuria. And thank you, Mark, and thank you, Tom. And now we have 15 minutes for you to discuss about the topics you are interested. And as you know, I came to you, and it seems that there are several things that came up already in your talks. It seems that, well, of course, you are very you overlap, okay, because the printing, the questions, whatever, 2 more academic and 2 more towards the citizens. But it seems that there is something common, is that, the point is, how can we have this safety, these ethical issues, how we solve the ethical issues? That is not only what Nuria was saying, because there are some ethics as well in the do-yourself biology movement, and how you solve that. And then how you can deploy the GMOs, the genetic modified organisms outside the labs, and how you see all that, let's start by this, and you can talk, since you are only 4, I think that there is no point that I give you the word. You can say your opinion about that. Who wants to start?
Well, let me maybe start, because this is an issue for our project in Peru, where we make genetic engineered organisms that we will use with the people. And so one of the things that we thought of doing is of making maybe cell extracts of them, so that you lyophilize the cells and then you just have cell extracts, and that's something that cannot spread in nature. Another thing that you could do also, but I'm not a specialist in that, is to use xenobiology, that you use some building blocks, artificial building blocks that these organisms need. So from the moment on they're out in the open, they die. So that's also something that you have to realize. The bacteria, for instance, that we use, they need a lot of supplements. And it's very unlikely that in nature they will find all the supplements in order to survive.
Yeah, but the point is that we complain because we don't like the soya, that is genetically modified.
I like it. So
but I mean the movement, you may like it, but most of the people, or let's say that many people, argues that it's very dangerous to introduce this in your fields.
That's exactly, I think, what's interesting about what Nuria is doing and what I'm doing, and a lot of those DIY biologists, is that you make it accessible and that people can think about it and get exposed to it and have a better feeling about it. Because the corn that we are eating, if you check that to the normal corn, or for instance a banana that we're eating, and you check that to the normal banana that was originally in nature, that's also changed. But that's changed by also by selecting all ways. So a lot of things that we are eating is already a product of genetic engineering. So, but I do understand that people have their doubts, and of course, but I do believe that they should be informed better. And that's why those open labs are interesting, because you can inform people better.
Yeah, that's a very good point. Oh, sorry. Go ahead, Nuria. Okay. No, actually, I was going to point some of the technical issues that Tom raised before, right? I think we are learning some that actually we have quite safe ways to use GMOs outside of the lab. For example, we can learn from the clinical trials, clinical testing of modified bacteria that have been done in the US since, I think, 2006 at least. And definitely using these auxotrophies, basically making these bacteria basically that they miss a key gene for processing certain metabolites, for example, DNA bases or amino acids. This makes them, of course, incompetent to survive outside of the lab. So I think that's a relatively well-tested technology. Of course, we need to keep testing and monitoring well, but we have, I think, relatively good solutions for that.
Nuria, you were going to say something, Nuria Montserrat?
Yes, it was in the same direction as Mark. So we can modify or abrogate, shut down specific enzymatic activities, or just repurpose the platforms for not being like having these kind of issues. And also we need to take into consideration that some of the tools that we are currently using in the lab, as for example ourselves using these cellular scaffolds, are currently used in the clinics. So they go through a whole process. I try to put a timeline for people to also think about this. So most of the things that we do in the lab are work for many, many years. It's work, investing first how things work, how to get cells out of a tissue, how to work with several scaffolds, and then it goes into a whole process of ethical review by local committees, national committees, and even at the European level, we have a lot of bodies that are regulating our work. So I think also these people need also to take into consideration that whatever project we run with pluripotent stem cells or human embryonic stem cells, they go through a whole process of review and through a whole process of debate together with the committees that are reviewing our work. I think the major issue here is when people feel that we are manipulating or manufacturing or something that we see with good eyes, and we are very used to that. Some people can get scared about this, and the good thing to teach or to educate people is by doing what Nuria or Tom are currently doing, like having access or giving access to people to explore their cells, but also putting some good instructions about how to do all these things and to put everything in open access for people to have access to these protocols and to these procedures. Something that in 3D bioprinting is also a standard, because we can be using designs from many labs using the open softwares for doing our designs, the protocols. So I think that this in the last 5 years is changing a lot in the field of 3D bioprinting. For example, this is a must. We are using open softwares for 3D bioprinting. We are uploading our software, and in the software as well, the designs. So this is also something that is helping a lot. But I think that most of the question that goes into how people deploys this into the field, no, this is something that for us is very normal. But of course, people, they need to be trained in that.
Yeah, no, but the point was it's a bit contradictory, no? Because in one side, in academics, what you need is to pass several ethical committees to be able to work within the lab with a very contained laboratory and to do all that. And then you have open space labs, no, like these biohackers or do-yourself biology, where these confinements are not approved, no? It's a kind of debate. And then on the other side, the society is very much concerned always about these modified organisms, no? That's a bit, in my opinion, the debate. But this is, of course, I think it's very important to stress that most of the things that are done mainly in academia, no, they pass these ethical issues, because now we will come to the CRISPR, Mark, and I'm sure you have many comments there. That's a fantastic technology that allows every country in Africa to edit genomes, no, of, I don't know, of the mosquito that transmits the malaria, and opens really new avenues for them. But at the very same time, CRISPR opens many debates, no, because, or as you were mentioning as well, Nuria, you can edit the germline, no? How do you see this? And Tom and other Nuria as well, you can enter there.
Well, the first thing that I like to bring to the conversation that's not very well known is that in USA, in the United States, FBI is actually doing, it's collaborating with a lot of the biohacking spaces in terms of the ethical issues and also the safety. So instead of, I don't know why this happened, but instead of going against and being like afraid of that, they embrace the idea and they make sure that this meets the security standards of FBI in that case. Also to see that the logo of DIY Bio is not a microscope or a Erlenmeyer or a pipette. The real logo, it's a goggles, it's a safety device, because it's always, or at least at the beginning, the movement was born together with the Cyberpunk Manifesto. So like it's a movement that comes from the ethics and says that the people needs to be educated and to have all the knowledge needed to take an informed decision about this kind of issues, especially the GMOs. And it's true that this is the most asked question. What happened with the GMOs, and the people it's actually very worried about that. I also have to say, and you know, because you are all scientists, that release a mutant to the environment and maintain that mutant in the environment is actually much more difficult than succeed in that. One thing is the invasor species or the crops that are in a field that they have a farmer taking care of them. But if you release a mosquito that doesn't have the right technology, like the drive technology, gene drive technology behind, it's actually very difficult to maintain the mutant in the ecosystem. Actually, the bioremediation technology has actually not worked very well because the microorganism, once it's modified to do something else for you, it's less capable to be in the environment and they die out. And you have to be always adding up this mutant all the time. So
actually, I remember what you mentioned, Nuria. I asked once to Victor De Lorenzo, actually one of the best environmental microbiologists in the world, exactly this question. And he said, well, we shouldn't worry. Actually, it's so hard to make them survive in the wild, whatever we touch in the lab. So
the message that I give to the people too, because they don't know.
But and also, yeah, in our movement, one of the first things they started to do is write ethical charters. And when we get together in the Bio Summit, there's a huge part about bioethics. Also interesting to know is that most DIY biologists have a biology degree. So they know what they're doing, and also for us, we exclude working with pathogens. So that's something that we simply don't do. We work with a E. coli that's not harmless. We work with normal yeast, so that's the only two microbes that we confine ourselves to. And also we're part of a school. So this is something that puzzles me, that you don't see a lot of open biolabs yet at schools. I think it's a very nice educational tool. We even implemented it in a program. It's not a mandatory course yet, I hope, for one day, but it's an extra course. People can have the freedom to experiment in a lab, in a safe environment with mentors. So that's very important to stress. There are some weirdos in the community, but that you have in every community, and they get a lot of airplay. But the majority really are people that are they love biotechnology. They want to make it accessible and understandable for everybody.
Yes, I think that the bottom line that in all communities, there is self-ethics restrictions, and with CRISPR has been very clear because scientists were the first ones to bring up the problems that this would imply, and to put these self-restricted measures. Of course, as you said, crises are everyone in all communities and in all societies. But I think it's very important for everybody to see that when they ask, scientists, what are you planning to clone someone? No, does not have much interest. Besides, it's very difficult, but it does not have much interest to clone myself. We don't care about that, no? But the point is that I think all communities that do have this kind of scientific criteria are pretty well self-organized in terms of ethics, no? And then what you said, Tom, is interesting because you mentioned or you brought up the mentorship, no? How you see this movement on open labs, the figure of the mentor, the importance that it has.
Yeah,
to empower people or whatever.
That's what we spend a lot of time for that. So whenever someone comes to the lab, we don't just open the lab and let them freely work. So they first have to tell us what they want to do, then we discuss how they're going to do that, and they get a one-by-one mentor. Also, we really try to guide them in the best way as possible. Also, one thing, as I showed you, we develop some workshops so people get introduced with the devices. If you do an Agger Art workshop, for instance, it's a cool way on how to learn. You learn how to grow your microbes, how to make plates, how to work with a micropipette. So by doing those things, they get a lot of new skills. And then, of course, in the lab, we always mentor and guide them and see what they're doing. So we don't let people just in the lab leave and then come back after a few hours to see what they've been doing. So you see, we're there all the time also.
There is a comment from the audience in the line that you were saying that, they were saying that although now most of this genetic engineering is done in the labs, many years ago already people working in agriculture, they had done this other kind of engineering, like grafting, two different kind of trees, and then you have a new species like nectarines and things like that, that in agriculture, this has been done in other ways, not in the labs, but mainly in the fields. Now, I think it would be nice if people of the audience take advantage that you have these 4 fantastic people here, and you have to squeeze them and ask them all the things you wanted to know and you never dare to ask. And I really invite you to do so. If not, I will take advantage, and I would like to discuss with you, because I'm really interested in this thing you already commented, the 100 Euro lab, that there are several papers that have been published in that matter, And I think it's interesting because in a way, this movement has entered as well in academics, because I'm sure Nuria, Mark, and myself in our labs, what we are doing as well is we always have people that they do 3D printing for us, that they print the rotors, or that they invent new devices, to optimize the things we need in the lab. Because, of course, up to now, we had all the things we're ready to buy. But now it's true that the multidisciplinarity makes that maybe we need devices that they are not even in the market, And I think it's nice because in a way that seems that are two segregated fields, like do-yourself biology, you are cool, and you are in the lab, you are not as cool because you are a classical scientist. It's nice because I think they are pretty much mixed. I don't know if you feel like that, or it's just me that I was born before the men arrived to the moon, and I'm seeing the things in another way. Sorry about that. You can comment on that.
For example, from our side, being a stem cell biologist, I don't know if I will look not cool or yes, but what is true is that we are needing a lot of technology-driven research. So we need to be developing tools or even devices like microfluidic devices to phenocopy the microenvironment that the cells do have in a process of differentiation. And then you can go into something very picky or very expensive, like entering into a microfabrication room and doing a microfluidic device, or you can talk with Nuria Conde and she will help you. For example, we were discussing about this in Heidelberg before the pandemics, you remember? So we do have a lot of interactions, and I think it's important, the dialogue, because you mentioned something about cool or not cool, but I think it's also important the training that we have at the daily basis, that perhaps we need to kind of approach more our work and to go more from academia back to open labs and to do this exchange in a real manner, because it can happen that we do not understand, though we are doing very similar things. And it's true that developing tools and devices for us is super important, but sometimes we cannot even understand each other what I'm really needing. And perhaps they already have this knowledge that happened with Nuria, like we were discussing how to approach something. So this is important, and we need to do this, as also Tom was commenting, by education and by training and by doing more things together. So totally right. We are needing to approach more our both, not disciplines, but perhaps way of thinking, or how we do train also the students to have this more open view of things and to share more knowledge, specifically knowledge, because from very old time, right, academia was looking more something like something closed that we are not so open to share, and this is not true at all. So the same happened with maybe open labs, that maybe many of you, like me, we think that open labs are constantly open and sharing, but perhaps it's not also true. So we do need to maybe break some dogma and some way of thinking to work all together in this. So I think multidisciplinarity is, as I mentioned in my case, being a stem cell biologist by training and entering into bioengineering has also changed a lot my way of approaching science, and it's being very challenging because I need to break a lot of barriers. So this is something that it needs to be overcome by young people.
Yeah, I also agree with Nuria that for me it's easier to find a super interdisciplinary environment in the DIY community, in the sense that you have an artist and you have a nanotechnologist, and all the conversations that she was referring to, I actually learned in the DIY bio because one of the other participants of the laboratory was a nanotechnology engineer. So he explained me how to do these chips, and then when I was in the Bio Summit with other biohackers, they did a workshop about how to do the microfluidic devices, and now I got that skill that I learned in a middle of a corridor and in the street. And it's not as professional as Nuria needs for the stem cells, but it gives you the platform to actually understand the high-level technology and to have the conversation with the people who is actually the specialist on that. Otherwise, for me, it's difficult to understand all the specific words, but in science, you need the specific words to actually make it understandable. But it's, and I think science is learning that. I think in the PRBB or in the IBEC are places that now we have a lot of this kind of people, but that's my personal experience.
Because, for example, in terms of workshops, it's something that now it's in most of the modern institutes of biology, right? You usually have a workshop, right? Which probably some decades ago was not available, right? I think EMBL definitely has had a workshop since many years. Now in the PRBB, it seems we will have one soon. So yeah, I think definitely this synergy is very clear that it's I don't know, more and more important.
And do you think that, in biology, well, that's what I know, biology or biotechnology, it has been an exponential growth in generating knowledge that was very much accompanied to the exponential growth in the development of new tools. Do you think that this will be similar in the open space labs?
Yeah, for
sure. That is at the beginning, and maybe this is going to be in the very same way?
Yeah. But because it's already happening. For instance, the sequencing to know how DNA is built up, that's becoming so cheap. Those devices, you have them and they're smaller and cheaper than an iPhone. So for just a few thousand, now it's maybe, I think, about 50,000 euros still to sequence the human genome with those new devices, but that will become cheaper and cheaper and cheaper, so that also it becomes more accessible for those labs. So we started, well, a lot of them started building those devices as DIY devices because they couldn't afford the real equipment, but that equipment is also becoming very, very cheap. For instance, you have a bento lab, which is for 1,500 euros. You get 3 different devices, 1,500 euros. People pay that for an iPhone, but you can also buy a centrifuge, a gel electrophoresis system, and a PCR machine for the same amount of money, and you will have 20 years of fun. So that's really happening. Those things are becoming cheaper, and also so people can use them. And I hope also they will start using it in high schools, because really I don't know how it's in Spain or in Catalonia, but in Belgium still they're not doing PCR. I don't understand this. I think when you're 16 years old, you should have done a PCR in school, so that you at least know what this is. And in the beginning,
now everybody knows what the PCR is, with the pandemics.
Yeah, but they need to know.
Either you are positive or negative.
They should know. And in the beginning, it's too expensive for schools, but now for a bento lab, it's 1,500 euros. Come on, we're not going to complain about these costs anymore. So it's a fake argument right now. So we're also helping schools on how to use that. So we're also building some workshops some easy to grasp things so they know how this will work.
I think that's very good. This is very interesting. And just we have 5 minutes, but I want to ask you something, because people in this movement, you always talk about empowering citizenship and have fun to educate people, whatever. Nuria already heard me to say that. But my question is, I think that this could be a revolution. But it seems that there is no one that wants to make a step forward because, I think that this can be a revolution because someone could be building these PCR machines and sending to all African countries that they could do a PCR to test if they are positive for AIDS, etc. No? But this seems to proliferate a lot in the first world country. Why?
I think that there is every time and every day there is a new DIY bio especially in it. I know the case of India because we have been working with India instead of Africa, and my experience in that is that they like to do the DIY bio together with the accelerators of a startup. They are kind of very practical people in the developing world. They want to have something not for fun, but because they need it, they really need it, they need it to work. So for me, the synergy is in the sense that now here we have DIY spaces that can do machines, that are already machines that are sellable, the ones that Tom was talking about. And then we are now sending this knowledge to Mexico, for example. Also, this is happening to Mexico. Peru are the ones that are collaborating with Tom. And in India, I know for sure that the BioReal, it's one of the most important accelerators of biotech technologies and started as a DIY. So I guess you are right, it should be done, but we are still in the first steps. My hope is that it will happen. It is hope.
Yeah,
I think.
But it's also developing something takes time and also takes money. You cannot single that out, and especially when you work with volunteers, they're passionate about it, but they're still volunteers. So our project in Peru, we got funding so we could hire some people in Peru that are working on it. And we hope that will be a start, that this will be a project with which we show to the people, look, biotechnology is useful. It can help you with something. And we hope that it will evolve, of course, into citizen science and then maybe set up small DIY labs. So we really hope this, but I think it's really important that you show that with a key example. So, and that's what we're putting a lot of effort in right now.
Okay. You want to comment anything else? If not, I would like to thank you very much for the effort to share all your knowledge with everybody, with all of us, and for this discussion. And we passed the session. We can close it here. And again, thanks a lot for the participation. And clap, 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 clap.
Thanks a lot, Cristina.
Yeah,
Thank you very much.
To
It was a pleasure.
everybody.
Yeah. Bye.
Ciao. Adeu.
Ciao.
Otro día más aquí, Maker FAIRE Girona, Visiting Spaces, un gusto como siempre acompañaros. Soy César García, estoy haciendo de maestro de ceremonias y vamos en esta Maker FAIRE Girona de un sitio a otro como si nos estuviéramos teletransportando para conocer los últimos avances en innovación, en ciencia y tecnología. Acabamos de ver este panel sobre toda la biología y cómo podemos acercarla a nuestro día a día, y justo tenemos la suerte de, en esta sección de FAIRE Girona, de contar con Rafael García, que es catedrático del Departamento de Arquitectura de Computadores de la Universidad de Girona, que nos trae una charla que a mí me da mucha curiosidad. Vamos a viajar al fondo del océano para entender mejor la lucha contra el cáncer. Rafael, ¿qué tal? Buenas tardes, bienvenido a Maker FAIRE Girona.
Hola, ¿qué tal? ¿Cómo estáis?
Pues nada, aquí muy expectantes de tu presentación, vemos ya tu pantalla y nada, vamos a comenzar ese viaje al fondo del océano. Así que
perfecto. Pues muchas gracias, César. Hola a tots i a totes. Bueno, començarem aquesta presentació. Fixeu-vos que anirem des del fons de l'oceà fins a la lluita contra el càncer de pell. Com comentava en César, jo soc un investigador, soc professor en el Centre d'Investigació en Robòtica Submarina, que és un centre que tenim a la Universitat de Girona, en el qual una de les coses que fem és desenvolupar tecnologia per explorar l'oceà. Tots aquests robots d'aquí són robots made in Girona, que hem fet nosaltres, i que es caracteritzen precisament perquè tenen molts sensors i que són capaços de fer coses de forma autònoma en el fons del mar. Una de les coses que normalment fem és anar a una determinada àrea del fons del mar, en aquest cas del litoral, amb aquest robot que és l'Esparus, però en tenim d'altres, com heu vist abans. Aquesta és la nostra embarcació, amb aquest pont grua que ens permet posar els robots a l'aigua, i normalment el que fem és programar aquests robots per demanar que vagin a explorar una determinada zona. Llavors, aquests robots el que fan és enfonsar-se i quan arriben al fons del mar, normalment exploren de forma relativament intel·ligent, és a dir, si troben un obstacle enmig de la zona que els hi hem dit que explorin, doncs són capaços de sortejar l'obstacle i van agafant imatges i van agafant tota una sèrie de dades que després o bé processen el propi robot mentre el robot va capturant aquestes dades, o bé quan torna a la superfície ens dona totes aquestes dades i aquestes dades ens permeten treure conclusions, i aquestes dades les compartim amb biòlegs marins, amb geòlegs marins, amb arqueòlegs, i d'aquesta manera tenim informació sobre un entorn, en aquest cas una pradera de Posidònia o una zona que té coralls de tipus Gorgònia o coralls d'un altre tipus, o veure si alguna determinada espècie invasora està ocupant una determinada zona. Llavors, això que veieu aquí és un tipus de mapa que és el que nosaltres acabem generant. Si nosaltres al cap d'un temps tornem a la mateixa zona i tirem el robot a l'aigua, el robot fa una trajectòria diferent, però fa un altre mapa. Aquests mapes els podem fer en 2D, com esteu veient aquí, o els podem fer en 3D, com és el cas d'aquesta col·laboració amb el Centre d'Arqueologia Subaquàtica de Catalunya, en el qual, veieu, en aquest cas és un vaixell del segle I abans de Crist. És un vaixell que es va enfonsar a prop de les costes de fet, a la Costa Brava. I fixeu-vos que amb les imatges que nosaltres agafem amb aquest robot, doncs això ens permet construir models de molt alta resolució que pràcticament són un registre visual permanent de com estava, en aquest cas, aquest mapa, aquest vaixell o aquesta pradera de Posidònia o aquesta zona de coralls, i veure si al cap d'un temps tornem a la mateixa zona, fer un segon mapa i llavors comparar aquests dos mapes. Llavors, la meva recerca, fixeu-vos que té a veure amb això, amb col·laborar amb arqueòlegs, amb col·laborar amb biòlegs. Fixeu-vos un altre exemple, en aquest cas, una col·laboració que fem amb IFREMER i amb investigadors de l'Institut de Físic du Globe de París, que són geòlegs, que estudien aquestes xemeneies hidrotermals. Això d'aquí no està agafat amb els nostres robots, està agafat amb un robot d'IFREMER, però nosaltres agafem aquestes imatges i també les processem. I amb tot això podem generar aquest model 3D. Això d'aquí és una xemeneia hidrotermal que hi ha a 2.000 metres de fondària on s'ajunta la placa tectònica d'Europa amb la d'Amèrica. I allà hi ha hidrotermalisme, per tant hi ha uns quants volcans submarins, i llavors és molt interessant poder veure de quina manera les condicions que es donen en aquest entorn, que en un principi ens pensàvem que no tindria vida, fa 40 anys tothom pensava, a 2.000 metres de fondària, com que no pot arribar-hi la llum del sol, no pot haver-hi vida en forma de planta i, per tant, no es pot sostenir la vida. Però si mireu aquesta zona d'aquí, fent zoom en el nostre mapa, us veureu que aquí hi ha músculs. Tot això marró, aquestes taques marrons d'aquí, són músculs que aprofiten l'aigua calenta que surt d'aquests volcans a molt alta temperatura, 300 graus, i a partir d'aquí, en lloc de fer la fotosíntesi, fan la quimiosíntesi. Nosaltres el que fem és treballar amb aquests geòlegs i amb els biòlegs marins perquè volen entendre de quina manera funcionen aquests ecosistemes extrems i de quina manera això evoluciona en el temps. Torna a ser el mateix. Anem un any, fem un mapa, tornem al cap d'un temps, fem un altre mapa i comparem aquests mapes i diem, mira, veus, aquesta zona d'aquí estan sortint plaques de bactèria i en aquesta altra zona està sortint aquest altre tipus de... o s'està produint aquest canvi. Llavors, tot això, aquesta tecnologia, no només és tecnologia que podem utilitzar en el fons del mar, sinó que jo, personalment, doncs resulta que, com aquest pacient d'aquí, tinc moltes pigues. I com que tinc moltes pigues, una de les coses que vaig començar a fer fa molts anys és anar a veure el meu dermatòleg. I el meu dermatòleg què feia? Doncs primer anava amb un dermatòleg que el que feia era agafar una lupa i mirar-se aquestes pigues. Però clar, si en tens moltes, doncs, com expliquem aquí, això és molt lent. És a dir, si ha de mirar cadascuna d'aquestes pigues, en segon lloc tenim un problema, que és que no hi ha una referència de com era aquesta piga fa 6 mesos. I resulta que ara me mira i diu, aquesta piga és sospitosa, què fem? La traiem? No ho sé, ha canviat o no ha canviat? Si ha canviat l'hauríem de treure, però si no ha canviat, no. I llavors em pregunta, tu creus que ha canviat? I jo què sé, la tinc a l'esquena, jo no me la veig. Llavors, és difícil percebre aquests petits canvis, no? I una solució que existeix avui dia és una cosa que es diu fer mapes manuals amb dermatoscòpia. I això vol dir que s'utilitza un aparell, que és aquest aparell d'aquí o aquest que teniu aquí, que és un aparell que té llum polaritzada i una càmera, i llavors el metge el que fa és piga a piga, posa l'aparell en aquella piga i fa una foto. I llavors té un software a l'ordinador que diu, mira, aquesta piga està en aquesta zona d'aquí. Però clar, quan tens un pacient que té moltes pigues, només per monitoritzar les que monitoritzaven a mi, que són aproximadament unes 50, quan en realitat en tinc 400 i pico. Doncs d'aquestes 50 que monitoritzaven, clar, estàvem entre 35 i 40 minuts per poder fer aquesta monitorització i veure la imatge de l'exploració anterior i com és la imatge en la següent exploració, l'exploració actual. I llavors, una cosa que vam dir amb el metge que em portava és, home, i per què no intentem automatitzar això? Perquè, clar, al final, trobar quina és la piga de les 450 que tinc, que pot haver fet un canvi, és com trobar Nemo en aquesta imatge d'aquí, no? Realment és un problema on les dimensions de l'espai de cerca i de les coses que poden passar doncs fa que haguem d'aproximar-nos d'una altra manera. I jo vaig pensar, bueno, doncs anem a fer-ho de la mateixa manera que ho fem en el fons del mar. I això és una col·laboració que tenim amb l'Hospital Clínic de Barcelona, amb la unitat de melanoma, i amb una empresa spin-off del nostre grup de recerca, que és Coronis Computing, que vam construir aquest escàner d'aquí. Això d'aquí és una cabina a la qual hi ha tota una sèrie de càmeres, un sistema d'il·luminació amb llum polaritzada, i aquí el que fem és col·locar-hi el pacient. El pacient es col·loca sobre una plataforma rotatòria, es posa en aquesta posició d'aquí i llavors el que fem és girar el pacient lentament mentre agafem imatges. D'aquesta manera, en 3 minuts, tenim una cobertura total del pacient. La metodologia d'escaneig és que primer té una posició a la que està mirant endavant, després, quan ha fet mitja volta, mira enrere i d'aquesta manera podem escanejar tota la superfície cutània del pacient. Això em permet automatitzar la construcció d'un mapa que després podré comparar amb la següent exploració. Val a dir que la qualitat d'imatge que tenim amb aquest escàner no és tan bona com la de la dermatoscòpia, però és molt millor que la que no tenia aquest mapa per detectar canvis. I fixeu-vos que aquí això m'acaba generant aquest model 3D del pacient en el qual tinc totes les pigues d'aquest pacient localitzades. Amb sistemes de machine learning detectem on estan aquestes pigues i sabem on en la superfície del cos del pacient estan aquestes pigues i llavors les podem col·locar per qüestions d'anonimització de les dades, les col·loquem en un avatar estandarditzat i aquest avatar estandarditzat és el que utilitzem després per comparar a la següent exploració i detectar aquests canvis. Per tant, això és tecnologia que ja hem desenvolupat i que ja està en marxa. Aquest escàner està a la unitat de melanoma de l'Hospital Clínic i està ja capturant dades d'aquest tipus. I veiem que tenim una qualitat d'imatge que no és tan bona com la dermatoscòpia, però és prou bona com per poder, com a mínim, detectar canvis. I a partir d'aquesta experiència, amb aquests metges que treballem del clínic, vam dir, home, i per què no intentem aconseguir un projecte més gros encara, que és un projecte, en aquest cas, d'aquesta convocatòria, que és l'última convocatòria del programa Horizon 2020 de la Unió Europea, que és la convocatòria Digital Diagnostics. Perquè us feu una idea de la dificultat d'aconseguir finançament en aquest tipus de convocatòria, fixeu-vos que es van presentar 158 propostes de projectes. Això és un projecte que vam escriure durant la pandèmia. Estàvem tancats i vam estar fent reunions virtualment amb tots els nostres partners, amb tots els socis del projecte, i vam enviar aquest projecte que es titula Intelligent Total Body Scanner for Early Detection of Melanoma. És a dir, anem a fer el mateix escàner que tenim, però millorant la qualitat d'imatge i fent que sigui molt més intel·ligent. És a dir, no només que utilitzi les imatges, que utilitzi més coses. Perquè us feu una idea, només es van finançar 5 projectes d'aquests 158 que es van rebre i el nostre va ser un dels projectes que es va finançar. Aquí la idea és que no només volem utilitzar la dermatoscòpia, aquestes imatges del pacient, sinó que, a més, volem utilitzar genòmica, és a dir, hi ha pacients que si analitzem el seu genoma veiem que encara que no tinguin moltes pigues, que això és un indicador que és un pacient d'alt risc, doncs té mutacions en determinats gens i sabem quins són aquests gens. Per tant, anem a estudiar els gens d'aquests pacients per veure quina és la probabilitat de que aquest pacient acabi desenvolupant un melanoma. I a més tenim l'historial clínic del pacient. Si jo sé que aquest pacient ja ha tingut 2 melanomes, el risc que té no és el mateix que un pacient que no n'ha tingut cap, per exemple. Llavors, utilitzant tota aquesta informació amb el que nosaltres anomenem un assistent cognitiu d'intel·ligència artificial, és a dir, un cervell electrònic que agafarà totes aquestes dades de tots els pacients i que em permetrà generar un model que prediu per cadascuna de les lesions pigmentàries de les pigues d'aquest pacient, quina és la probabilitat que aquest pacient desenvolupi un melanoma. Fixeu-vos que aquesta és una representació del tipus de dades que utilitzarem, l'historial clínic del pacient per un costat, en el qual tinc en compte si té familiars que hagin tingut problemes, quines són les característiques de la pell d'aquest pacient. No és el mateix un pacient amb la pell amb un fototip de pell molt clara que un pacient que tingui fototip de pell més fosca, no és el mateix un pacient que hagi patit, per exemple, problemes de cremades a la pell que un pacient que no ho hagi patit, i tot això és el que anomenem l'imaging phenotype. Quines característiques té la pell d'aquest pacient. I això ho combinarem amb aquest tipus de mapes, però de molta més resolució que farem en aquest projecte. I, per últim, tinc la part de l'estudi genètic d'aquest pacient. I amb tot això generarem el que anomenem aquest Holistic Risk Score, aquesta quantificació del risc de forma holística perquè tingui en compte tota la informació possible. Aquesta és una visió del consorci que hem muntat. Fixeu-vos que aquí no és un projecte que fem només, l'hem liderat i l'hem engegat i el coordinem des de la Universitat de Girona, però aquí hem implicat a 6 universitats, tenim 4 empreses industrials, Bosch, IBM, Barco i Rico, que també participen en el projecte, tenim diverses empreses que són petites i mitjanes empreses que estan especialitzades en fer coses o bé d'òptica, o bé d'intel·ligència artificial, o bé de pell directament, i tenim també els nostres partners clínics, que són l'Hospital Clínic de Barcelona, amb el que treballem, però també muntarem un escàner a l'Hospital de Trieste, a Itàlia, i tindrem un tercer escàner durant el projecte a Austràlia. Per què? Perquè volíem tenir pacients amb fototips diferents, amb característiques de pell diferents, de forma que els pacients que tenen a Austràlia, com que allà hi ha molta més radiació solar i a més són pacients que tenen la pell més clara, doncs tenen un risc de melanoma que és diferent del que tenim nosaltres i volíem complementar l'estudi d'aquesta manera. I penseu que cadascun d'aquests hospitals tindrà un escàner com aquest que us he explicat i reclutarà 250 pacients. En total farem un estudi amb 750 pacients que seguirem al llarg del temps per desenvolupar aquest model que que estàvem comentant per la classificació de pigues. Llavors, per acabar ja, bàsicament, l'aspecte de l'escàner que volem generar és una cosa semblant a això, en el qual tinc una sèrie de càmeres de molt alta resolució que em permeten focalitzar en el cos del pacient i de forma molt ràpida capturar totes aquestes imatges i combinar-les amb les característiques de l'historial clínic del pacient i de la genòmica per poder acabar tinguent aquesta estimació de quin és el risc de forma ràpida de que aquesta piga, en concret d'aquest pacient, tinguent en compte el seu historial i tinguent en compte la seva evolució i les seves característiques genètiques, doncs acabi desenvolupant o acabi sent una lesió maligna i, per tant, l'hem de treure. Penseu que en el cas del melanoma és molt important el diagnòstic precoç, perquè si agafem un melanoma quan encara el seu gruix és més petit que un mil·límetre, doncs la probabilitat de supervivència del pacient és altíssima, més del 95%, i en canvi, si és més tard, comencem a tenir mortalitats molt, molt elevades. D'acord? Doncs fins aquí la meva presentació i si teniu alguna curiositat, pregunta o hi ha algun aspecte en el que vulgueu que desenvolupem una mica més el tema, doncs estaré encantat de responder a vuestras preguntas.
Perfecto, Rafael. Pues muy interesante la presentación y muy inspirador ver cómo se combinan, ¿no? Y cómo las ideas de un área pueden llegar a otro lugar y a partir de ahí traernos descubrimientos totalmente nuevos. Aunque, claro, viendo, por ejemplo, todas las imágenes submarinas, se entiende muy bien la complejidad que hay en generar un mapa de algo tan complejo. Pero al final, bueno, nosotros también, en nuestros cuerpos también tenemos un montón de detalles y cuestiones. Una de las preguntas que, bueno, hay gente por aquí conectada en el chat, en YouTube, como siempre, animaros, si estáis ahora mismo viendo y queréis mandar alguna pregunta a Rafael, podéis hacerlo a través del chat. Vemos que Carmen Cesc lleva aquí, está abonado, lleva toda la semana compartiendo preguntas, pero de momento las estaba dando para justo el panel anterior. Quería preguntarle, Rafael, hablabais de un conjunto de unos 750 pacientes repartidos en los hospitales a lo largo del tiempo y quería preguntarte, porque bueno, ahora todo el mundo está emocionado con el Machine Learning, con las posibilidades de Machine Learning, y el principal reto en este caso es construir el modelo, evaluar el modelo, ver cómo funciona a lo largo del tiempo. Y te quería preguntar cuánto tiempo se tarda en tomar esos datos, ¿no? Entiendo que hay un estudio longitudinal que puede ser... O sea, ¿cuánto tiempo pasa desde que se llega a esta idea, ¿no? Hasta que es una cosa que ya encontramos en el día a día o que bueno, por lo menos que tiene la validación clínica de que el modelo es adecuado. ¿Cuánto tiempo puede pasar, más o menos?
Nosotros, de hecho, vamos a desarrollar este proyecto durante 48 meses, es decir, tenemos 4 años para el desarrollo del proyecto. Durante los primeros 18 meses vamos a estar diseñando y desarrollando el escáner, el nuevo escáner. En paralelo hay una serie de tareas que se van haciendo, pero en el mes 18 vamos a tener el primer escáner en el Hospital Clínic. Y ahí vamos a empezar a poner pacientes en el escáner, a hacer que haya pacientes que pasen por el escáner. 2 meses más tarde tendremos un escáner en Trieste y 2 meses más tarde tendremos un escáner en Brisbane, en Australia. Vamos a estar durante 2 años recogiendo datos con esos pacientes y de hecho, 2 de los partners que tenemos en el proyecto se dedican básicamente a hacer una de las tareas más tediosas pero que es imprescindible para utilizar Machine Learning, que es el utilizar los datos, anotar esos datos, pasarlos a los médicos para que puedan complementarlo con su diagnóstico y a partir de ahí generar el dataset que nos permite entrenar modelos de Machine Learning. Con lo cual, vamos a estar desarrollando el modelo y vamos a ir alimentando ese modelo con nuevos datos prácticamente hasta el 3er año del proyecto. Y a partir de 3 años y poco del proyecto vamos a hacer un segundo estudio que es un estudio de viabilidad, de análisis de viabilidad. Vamos a añadir nuevos pacientes que no habíamos visto antes y con el modelo que hemos entrenado vamos a intentar predecir y vamos a comparar lo que nos dice el modelo que hemos entrenado nosotros a partir de este escáner y lo que nos dicen los paneles de dermatólogos que van a estar validando el modelo. Con lo cual, fíjate que estamos hablando de algo que se va a dilatar prácticamente pues, 4 años en poder tener una validación del modelo y poder saber si es bueno o no para la práctica clínica. La manera como hemos escalado este estudio es permite hacer varias actualizaciones en el tiempo, de forma que a medida que vamos añadiendo más datos iremos viendo cómo iba mejorando la predicción del modelo. Pero es muy importante en un proyecto de este tipo el pensar en cómo vas a etiquetar los datos y tener toda una estructura de procesado que permita precisamente el generar toda esta cantidad de datos. Piensa que hemos hablado de 750 pacientes. De media, cada paciente le vamos a anotar por lo menos unos entre 50 y 150 pecas. Con lo cual, si haces el producto, acabamos con varios millones de pecas con su diagnóstico, con los datos del paciente, con la genómica, etcétera. Con lo cual, claro, la cantidad de datos que vamos a generar es muy, muy importante y va a ser precisamente una de las cosas en las que hemos puesto especial interés, es decir, bueno, pues cuando acabemos con estos datos, no nos los vamos a guardar. Estos datos los vamos a poner en un repositorio público en el que vamos a hacer lo que se conoce como un challenge. Es decir, vamos a lanzar un challenge en el que le vamos a pedir a todos los grupos que trabajan en Machine Learning en el mundo que se apunten y que participen y que compitan en generar el mejor modelo posible para procesar esos datos. Es decir, nosotros vamos a publicar qué es lo que hemos hecho nosotros. Oye, nosotros lo hemos hecho así, estos son los resultados que estamos teniendo, pero aquí tenéis los datos y ahora que todo el mundo, porque hemos visto en el pasado que este tipo de retos, este tipo de challenges, han hecho avanzar muchísimo los modelos de inteligencia artificial. Con lo cual, es muy importante para nosotros, en este caso, ya que tenemos unos datos muy valiosos que habremos generado a partir del proyecto, pues que esto esté a la disposición de la sociedad y que podamos hacer avanzar entre todos el diagnóstico del cáncer de piel.
Pues me parece una noticia 100% inspiradora. El que hoy en día todavía no esté haciendo Machine Learning tiene tiempo en estos 4 años para ponerse las pilas y pensar cómo mejorarlo. Por aquí te mandan la enhorabuena por el proyecto de José Boada y Jordi Freixenet, que dicen que están impresionados con el proyecto. Te dan la enhorabuena. Yo también te doy la enhorabuena por mi parte. Y hablando de tiempo, tenemos que seguir con esta tarde de actividades. Tenemos ahora una pausa programada. Rafael, muchas gracias por tu tiempo. La verdad es que es espectacular y os seguiremos la pista. Y deciros que ahora justo paramos hasta las 7:10. Vamos a estar con Núria Montserrat del Instituto de Bioingeniería de Cataluña. Vamos a seguir investigando todo lo que tiene que ver hoy con la biología aquí en Maker FAIRE Girona. Pero como os digo, no es ahora, sino 7:10. Hacemos una pequeña pausa, tomamos algo de agua, andamos un poco y volvemos aquí ahora mismo para seguir visitando espacios interesantes. Gracias a todos y un saludo, Rafael.
Saludo, gracias.
países, conociendo lugares interesantes, personas interesantes, investigando en ciencia, tecnología. Hemos visto de todo esta semana y justo para esta presentación nos vamos a ir a visitar un lugar muy especial. Nos acompaña Núria Montserrat, que es profesora de investigación y CREA e investigadora principal en el Instituto de Bioingeniería de Cataluña, que nos va a hablar sobre células madres para generar microórganos, un tema que es como, ¡pum!, ciencia ficción. Como siempre, vamos a empezar visitando los espacios, visitando los lugares y vamos a ir directos al laboratorio con este breve vídeo.
Benvinguts al Laboratori de Pluripotència per regeneració d'òrgans. El nostre equip es troba treballant a l'IBEC, l'Institut de Bioenginyeria de Catalunya, un equip multidisciplinar composat per biòlegs, biotecnòlegs, químics, on tenim l'obsessió d'entendre com es generen els teixits en el nostre cos per poder entendre també com, d'alguna manera, no només conèixer aquests processos tan fascinants de la biologia, sinó també com podem aturar o generar tractaments per malalties humanes. En aquesta sala de cultiu on estem ara, cultivem els nostres mini-òrgans i realitzem diferents assajos cel·lulars i també moleculars on podem cultivar aquests organoides utilitzant sistemes de cultiu, generant aquests petits microòrgans que podem no només visualitzar en el laboratori quan estan vius, però també després extreure el seu material genètic per entendre com els tractaments que fem servir impacten o frenen algunes qüestions que ens interessen relacionades amb la patologia del ronyó, amb la patologia humana. El nostre equip, com us comentava, treballa amb organoides, fent servir enginyeria, tal com l'estratègia d'impressió 3D i sistemes de microfluídica. Moltes gràcies.
Bueno, aquí acabamos de ver este vídeo con Nuria Montserrat. La verdad es que se os ve ahí manos a la obra. Justo tenía la duda de si las mascarillas siempre las llevabais o no en el laboratorio, o si era una cuestión COVID. ¿Cómo es esto? ¿Cómo es? Sí. ¿Cómo lo hacéis allí?
Lo hacemos, imagino, como ahora, desafortunadamente, en cualquier entorno laboral, que tenemos que tener la distancia de seguridad y trabajar, evidentemente, con estas mascarillas. Y como en el laboratorio hemos trabajado en investigación COVID desde el principio, pues tuvimos la suerte de poder seguir investigando con estos microriñones ya durante los meses de febrero, marzo, abril, incluso cuando hubo este cierre, ¿no? Este lockdown, en el laboratorio el instituto nos facilitó el poder seguir trabajando para investigar en COVID. Y nosotros llevamos con la mascarilla incluso antes de marzo, porque yo venía de Italia, fui a hacer un congreso y el fin de semana que volví a Barcelona casi, yo tenía la sensación de que no pasaba. O sea, pensé, mejor evitar, ¿no? Evitar ahora y íbamos pues, con mucha cautela, con mucha precaución en el instituto donde trabajamos. Entonces, las mascarillas ahora es nuestro día a día y trabajamos en turnos. La suerte es que tenemos diferentes espacios en el laboratorio, diferentes como si fueran salas de cultivo, salas de manipulación para hacer biología molecular. Entonces podemos estar muy poquitas personas trabajando a mucha distancia y lo bueno es que podemos hacer turnos pues largos, pero cada uno llegando a trabajar pues las horas que puede, ¿no? Porque también el COVID ha complicado nuestras vidas a todos los niveles. Pero sí, ese es nuestro día a día. Mascarilla, bata, por supuesto, y guantes, que eso siempre, pero ahora la mascarilla es
el día a día.
Tarea.
El elemento básico para arrancar. Justo nos hablabais de la investigación que estabais haciendo en COVID-19, también con el tema de microriñones. Y quizás yo no vengo del ámbito de la biología, la cuestión de los microriñones o qué son, cómo funcionan, si nos pudieras comentar ¿cómo ha sido esta investigación y cuál ha sido ese descubrimiento en el que habéis trabajado? Yo creo que sería muy interesante para la gente que nos acompaña ahora mismo.
Sí. Nosotros trabajamos con células madre, específicamente células madre que llamamos pluripotentes porque tienen la capacidad de, una vez sabes cómo trabajar con ellas, ¿no? Pues pueden dar lugar a los 200 tipos de células que conocemos que existen en nuestro organismo, ¿no? Esas células madre son células madre embrionarias que podemos aislar y poner en cultivo a partir de embriones humanos, de procesos sobrantes de FIP, gracias a un consentimiento informado previo. Estas células están depositadas en bancos, en lugares donde nosotros podemos tener acceso en contexto de investigación biomédica y también podemos generarlas en el laboratorio mediante un proceso que se llama reprogramación celular. El tener estas células, imagínate unas células que están en el momento cero, como cuando tú eras prácticamente un embrión, nos permite poder instruirlas, guiarlas, dándoles en el medio de cultivo esas instrucciones que muchas veces van a ser factores de crecimiento, vitaminas, que sabemos que facilitan pues, que estas células tomen una decisión, ¿no? Y que vayan hacia tejido de riñón, hacia tejido de corazón. En nuestro laboratorio estamos muy interesados en el tejido del riñón y en el tejido cardíaco, en el tejido del corazón. ¿Por qué? Porque durante el desarrollo estos tejidos se derivan, ¿no? de un tejido muy específico en el embrión. Y hemos trabajado mucho tiempo con modelos animales como el ratón y hemos podido entender cuáles son los genes, cuáles son estas proteínas, cuáles son estos factores de crecimiento que nos ayudan a instruir estas células. Trabajar con estos microriñones, ¿cómo lo hacemos? Imagínate una placa de cultivo en la que tenemos estas células en el momento cero de este reloj biológico. Empezamos a utilizar estos factores, estos cócteles, ¿no?, de diferenciación, y en 16 días generamos unos cultivos tridimensionales que son grupitos de células que se parecen mucho si las comparamos con tecnología muy importante como la secuenciación, por ejemplo, a nivel de lo que expresan estas células. Se parecen mucho estos microriñones a los riñones durante la gestación humana, en el segundo trimestre de gestación, en un proceso que dura únicamente 16 días. Es decir, algo que en el desarrollo tardaría 5, 6, 7 meses, en la placa de cultivo, en 16 días, podemos generar estas eso es importante, estas microestructuras que serían todavía muy pequeñitas, pero que las células se parecen muchísimo a nivel, como si fuera el fenotipo, ¿no? Como si fuera la pinta que tienen, se parecen mucho, ¿no?, a las células de un riñón embrionario. Entonces, trabajar con estos microriñones nos ha llevado mucho tiempo, pero hemos tenido que entender qué señales, cómo hacerlos, cómo generarlos. ¿Qué ocurre? La enfermedad del COVID, como todos sabéis, es debida a un coronavirus. Este coronavirus, que se llama SARS-CoV-2, utiliza una puerta para entrar en nuestras células. Hemos escuchado muchísimo de la mucosa nasofaringea, de los pulmones. Hay una puerta de entrada que se llama AC2, que es una enzima que convierte una molécula en otra, pero no únicamente está en las vías aéreas respiratorias. Esta puerta también está en los riñones, también está en el corazón. Nuestros microriñones que hacemos en el laboratorio, en 16 días, ya tienen esta puerta expresada. ¿Qué pensamos? En un estudio colaborativo con 2 investigadores, en febrero, tan pronto como en febrero, dijimos, hombre, si nuestros microriñones tienen esta puerta de entrada, podríamos generar un modelo de estudio de los primeros pasos de la infección de SARS-CoV-2. Trabajando en colaboración con un equipo austriaco que también hacían otros microórganos, en este caso como si fueran pequeños vasos sanguíneos, enviábamos nuestros microórganos a Suecia, en una sala de cultivo de nivel 4, donde habían propagado y cultivado virus de pacientes confirmados con COVID-19 en Suecia en enero. Y empezamos muy rápido, febrero, marzo, a infectar los organoides que hacíamos en Barcelona, en el Instituto de Ingeniería de Cataluña. Salían un lunes por la mañana en avión, llegaban el martes por la tarde a Suecia y empezamos a ver si, uno, este modelo funciona, realmente nos puede explicar alguna cosa, nos puede decir algo de cómo infecta el virus las células, y lo más importante, podemos hacer testar compuestos o moléculas que puedan frenar la replicación del virus, podemos intentar utilizar este modelo para algo más. Y tuvimos la suerte de que uno de los 3 que estaba liderando esta investigación, el doctor Joseph Penninger, hace mucho tiempo fue el primer investigador que descubrió esta puerta de entrada para la infección del SARS-CoV-1 en el año 2003. Él ya descubrió esta puerta, AC2, como una puerta de entrada para el SARS-CoV-2 y había desarrollado un fármaco que parecía que podía tener cierto uso para esas infecciones de coronavirus. Lo probamos en los microriñones en el mes de febrero o marzo, utilizar este compuesto. Vimos que este compuesto podía bloquear entre 1000 y 5000 veces la replicación del virus en los microriñones. Y hoy en día este compuesto se está probando en 6 países alrededor del mundo, no todavía en España, esperamos que muy prontito lo tendremos aquí, pero hoy en día ya están 6 países probándose en pacientes con COVID-19. Entonces, este fue nuestra primera aportación, ¿no?, desde el campo de la biología fundamental, de lo que sería pues entender cómo se generan estos tejidos, que a nosotros es una pregunta que nos fascina, ¿no?, cómo durante el desarrollo se genera un tejido, entender cómo se genera para después entender en un contexto de enfermedad, tener un modelo de estudio y después intentar entender cómo desarrollar algún fármaco para poder bloquear, en este caso SARS-CoV-2 infection, pero otras veces ha sido pues una patología de origen genético.
Aquí entiendo que todas estas medicinas que habláis, cuando vemos de vez en cuando, buscan inhibidores son precisamente cualquier tipo de barrera que pudiera bloquear esa entrada y de forma que, digamos, se impidiera que el coronavirus se extendiera. Me parece muy interesante, me ha quedado súper claro viendo todos los pasos. Me sorprende también el carácter tan internacional de la ciencia, ¿no?, hoy en día, ¿no?, como para llegar a este descubrimiento al final hace falta alinear muchísimos esfuerzos y muchísimo expertise distinto. Me decías justo antes de que entráramos que el campo en el que estáis investigando hace 5 años era como... O sea, está todo por hacer, ¿no? Y quería preguntarte, en toda esta cuestión de microorganismos, ¿qué microorganismos están, qué existe ya, digamos? ¿Qué se puede hacer?
Serían microórganos que se pueden generar a partir de estas células madre adultas, que hay mucha tecnología a partir de células madre adultas y nosotros hacemos a partir de estas células madre pluripotentes, ¿no? ¿Qué se ha hecho hasta ahora? Microcerebros, microcorazones, microriñones, micropulmones. Siempre, si partimos de células madre pluripotentes, esas que te comentaba que son las que aislamos a partir de embriones o que generamos en el laboratorio de manera artificial, lo que podemos obtener en un tiempo bastante corto en el laboratorio no va a ser igual a nivel de funcionalidad o a nivel de fenotipo, esto que te decía, ¿no? Como si fuera su aspecto, igual que en el tejido final, ¿no? Igual que un órgano final. Pero sí que en los últimos 5 años este campo de los microórganos ha sido un boom en el contexto de la biología del desarrollo o de la biología celular. Porque hasta ahora tenemos que pensar que durante muchísimo tiempo todas las cuestiones en relación a ensayar fármacos, las cuestiones que tienen que ver con probar según qué tipo de compuestos, se han hecho con cultivos en monocapa, con cultivos en 2 dimensiones. Los microórganos son estructuras tridimensionales que recapitulan muy bien cómo están las células dispuestas en el órgano, aunque sean embrionarios y aunque sean todavía muy inmaduros, y después no únicamente cómo están dispuestas, sino que podemos incluso recapitular o copiar o podemos ver que tienen alguna función, y eso es muy importante para poder formular estas preguntas. El boom de los microórganos fue reconocido, ¿no?, por una revista muy importante en nuestro campo, por Science, como el breakthrough, ¿no?, de los 10 descubrimientos más importantes, ¿no? Y yo tuve la suerte de participar en el primer estudio en el que hicimos microriñones, pero en este caso eran quiméricos, utilizábamos tanto células humanas como células de ratón, y en el campo lo que hemos hecho ha sido ir un paso más allá y hacer microriñones únicamente con células humanas. Y eso es muy importante porque nos permite formular estas preguntas en un contexto humano. Eso es importante si tenemos en cuenta la biología, cuando utilizamos animales, por ejemplo, para formular preguntas, o cuando utilizamos modelos preclínicos o modelos animales de experimentación. Ahora estaríamos trabajando con células humanas dispuestas en 3 dimensiones, estos microórganos, que nos permiten hacer unas investigaciones que decimos un poquito más aplicadas, ¿no?, un poquito más avanzadas.
Aquí estoy pensando hay un montón de información y cada más te escucho, más preguntas me surgen. Hablábamos yo, por lo que entiendo, todas esas pautas para ir organizando a las células pluripotenciales, entiendo eso como recetas, ¿no? Como unos momentos, hay que hacer esto, como unos pasos. Y hablas también de la parte 3D. Se ha hablado mucho también de la cuestión de la impresión 3D, de órganos, de tejidos, de cartílagos, y quería saber, en el campo en el que trabajáis vosotros, la impresión 3D, ¿qué aporta, digamos, o cuál es, o sea, a lo mejor es una herramienta más dentro de miles de herramientas o es el paso 500 de la receta, pero si nos pudieras decir la impresión 3D, ¿cómo se utiliza hoy en día para vuestro trabajo?
Has explicado muy bien, ¿eh? O sea, nosotros, en realidad, esta receta de cocina, ¿no?, o este manuario de cocina, no es algo que venga solo. Estos son más de 50 años. Incluso he tenido la suerte antes de explicar en otra presentación, ¿no?, de dónde viene esto de la biología del desarrollo. Tenemos estudios que en los años 1878, en los años antes del 1900, ya habían biólogos del desarrollo que querían entender cómo se formaba el corazón, por ejemplo, utilizando embriones de rana, ¿no?, porque podían mirarlos en una lupa, ¿no? Entonces, es importante tener en cuenta que estos estudios, ¿no?, los que estamos realizando en el laboratorio, tenemos primero toda esta fase de receta de cómo instruir las células para ir a la impresora y poder generar no únicamente 2, 3, 5, 10 organoides como hacíamos a lo mejor hace 5 años, hacíamos del orden de 100 organoides en un experimento, necesitamos a lo mejor a 2 personas. El entender estos procesos y poderlos sistematizar, poder hacer que este recetario de los primeros pasos esté sistematizado, nos permite después poder ir a la impresora 3D para poder hacer lo que se llamaría más el manufacturing, ¿no? O sea, poder hacer más organoides de manera más robusta, de manera controlada. En este campo, en el campo de los microórganos, hay muchas cuestiones, ¿no? Porque estamos generando a partir de células que son totalmente diferenciadas un microórgano. Y para nosotros, en el campo de la ingeniería, utilizando impresión 3D, lo más importante o donde estamos viendo que hay un mayor freno, pero al mismo tiempo una mayor investigación, es en la biotinta, que utilizamos junto a las células para hacer la impresión. Utilizamos, por ejemplo, impresoras de extrusión, ¿no? Donde vamos a poner las células junto a un biomaterial, un gel, un hidrogel, y vamos a imprimir las células con este material. Claro, tenemos que estar seguros de que una vez hayamos imprimido, lo que estemos imprimiendo, este constructo, pueda sobrevivir. Es material vivo, son células que van a estar en el camino de ser un microrriñón. Tenemos que saber en qué momento vamos a hacer este proceso de impresión. Tenemos que saber cómo generar estas biotintas. Entonces, en el campo nuestro hay mucho desarrollo alrededor de lo que sería generar estas biotintas, ¿no? Estas tintas biológicas. En nuestro laboratorio, en el IBEC, trabajamos en generar biotintas que decimos de origen biológico. A partir de tejidos humanos o tejidos de cerdo, podemos sacar las células y generar geles que nos ayudan a instruir esta diferenciación. En el campo, por ejemplo, como comentabas, del cartílago o del hueso, la aproximación a veces es distinta. La tinta puede ser una tinta que no sea de origen únicamente biológico, sino tintas sintéticas que además tienen un potencial incluso inductor, un potencial de regeneración, de autorregeneración. Entonces, en nuestro campo hay poquita cosa todavía hecha para hacer impresión de organoides, impresión 3D de organoides, pero estamos muy focalizados como campo de investigación en lo que serían estas biotintas. Uno de los objetivos, poder generar estos microrriñones del orden de cientos a miles y poder hacer estos estudios que decimos más de medicina de precisión de manera más robusta, ¿no? Que no dependamos tanto de una manipulación manual en la que sabemos que vamos a estar incorporando un montón de errores que no vamos a poder controlar, ¿no? Claro, estos procesos que se llaman de estandarización llevan muchísimo tiempo, pero sí que es verdad que en los últimos 2-3 años vamos encontrando más trabajos en cómo generar estos microórganos utilizando impresión 3D.
Esto es, hay que dar la bienvenida también a los robots, que nos echen una mano y que siempre pongan precisamente cada cosa en su lugar. Pues Nuria, se nos va acabando el tiempo. La verdad es que ha sido muy interesante saber pues todo lo que habéis trabajado. Antes he dicho microorganismos, no sé por qué, estaba entre microorganismos, la parte de organoides y órganos.
Microorganismo, ¿no? O sea, nos sabremos al final
Y darte las gracias por tu tiempo, por acercarnos, bueno, pues todos estos avances para que toda la gente que está ahora mismo viéndonos aquí en Maker Faire Girona sepa, pues, un poco dónde está la frontera y dónde están los siguientes pasos. Y nada más, darte las gracias y dar paso a los siguientes artistas, que todos los días acabamos aquí con una performance, acabamos aquí con una actuación. En este caso, hoy, último día de Maker Faire Girona, vamos a estar con Lolo y Sosaku, que son dos artistas que investigan las posibilidades de la escultura como terreno amplificado. ¿Qué significa esto? Pues todo lo que hacen tiene que ver con movimiento, con pintura, con formas de trabajar, con embeberse en la tecnología y desde ahí generar. Así que no me quedan muchas más palabras. Gracias y agradeceros a todos que nos acompañéis aquí en este Visiting Spaces esta semana de Maker Faire Girona. Si os ha gustado, compartidlo con vuestros amigos, conocidos. Si tenéis un momento favorito, ya sabéis, se lo mandáis. Mira qué interesante esta cuestión de impresión 3D, los submarinos. Hemos visto de todo en esta semana y hemos acabado aquí en el IBEC con Nuria Montserrat. Me despido por hoy y nos veremos en breve. Así que un saludo y vamos ya con Lolo y Sosaku.
Gracias, buenas